I'll first introduce Leighton, Howells, and Kyla Teplitz will also speak. Kyla is trained as an engineer, is now a senior product manager at RareCyte, focusing on making cellular imaging products, specializing in high-resolution fluorescence imaging. And Leighton Howells is the vice president of business development at RareCyte, and he has extensive experience in multiplex tissue imaging technologies. So excited to hear what they have to say.
Great, thank you, Sandro. Thank you, Peter. We certainly appreciate you pulling this event together. We know how much work it can be, and also the team behind the scenes of Madison and Chris and others. We appreciate all your effort today.
There's one thing I forgot to say, Sandro forgot to say, by the way. This event is being filmed. It's not being live streamed, but we did get agreement to film it, and it will therefore be available subsequently for review and made available to the community. But actually, we should have had a notice up to let you all know that. My mistake, sorry.
Okay. So everybody smile.
Actually, only Leighton is being filmed. The rest of us.
Well, thank you, Peter. That's good to know. So yeah, it really is our pleasure to be here. We've been working with Ultiview and Indica for a little while, so hopefully what we're going to bring together today is a story across imaging, analysis, and deep review with really enabling reagents. So we're going to try and bring that story together for you this morning, and then we'll pass from one to the other as we go. So at RareCyte we really have a focus on oncology. It's a very sharp focus, and when you look at the statistics, it's fairly clear why that is. US last year, 2018, there were 1.7 million new cancer cases. There were nearly 700,000 related deaths. When you take that to a worldwide scale, it's roughly tenfold that. So, this really is a global epidemic that is very significant. And while there have been, incredible advances in immuno-oncology and precision medicine, clearly there's still much more to be done because the actual rates of mortality are not declining for many of the major indications where early screening is not involved. So in my part of the presentation, I'm going to talk about two things, really. I'm going to talk a little bit about liquid biopsy, and I'm going to try and connect that to tissue for you. And then we're also going to talk about our tissue platform and start to show you some of the early applications work we've been doing with the Ultiview reagents. So RareCyte has a vision to really enable the next generation of analysis for rare cells and tissues for both research and for diagnostics, so for patient outcomes. And that's our liquid biopsy piece. And really this company has a very deep experience and expertise in, let's call it high performance imaging, really imaging at the leading edge in high resolution and super resolution. And then also a very clear past in a previous company around high content screening systems. And it's that expertise and that capability that cuts through into our current platform, not only in the instrumentation, but also in the engineering behind the scenes in terms of sample preparation, et cetera. So what we're going to focus on a little bit is some rare cell analysis first, where we have an end-to-end platform to identify cells in patient blood, to phenotype them with a number of markers, and then also to retrieve those cells. And that cell retrieval cuts across both single cells in a liquid biopsy and then across tissue microregions in a tissue sample. Our platform is utilized worldwide in academic centers. We're very pleased that Peter is a close collaborator along with others here in terms of pushing the boundaries of our systems and where they can go next. We have ongoing clinical trials and diagnostic relationships with many big pharma accounts. We're actually based in Seattle, downtown Seattle, where we have a really nice facility that supports our application development, our product development and manufacturing. And really the key message here is that we're building up that infrastructure to enable the quality and regulatory capability to support companion diagnostics for pharmaceutical companies. So what you may have interpreted then is we're talking about one platform to go from discovery to diagnostics. And the belief that the same platform, the same high-performance imaging, the same types of user interfaces, in some cases the same type of reagents cutting through is going to be a significant advantage, particularly for anyone in that translational space. So on tissue imaging, it's high-performance multiplexed immunofluorescence, as well as a bright field for H&E and IHC. automated high-resolution whole slide scanning. It's very fast. We'll give you some stats a little bit later. And then the microregion retrieval that's really important in terms of deeper investigation. On the cell imaging side, it's the same story. It's still multiplexed immunofluorescence, but then added onto that is the capability to take patient blood, extract nucleated cells, cancer cells or CTCs, spread them onto a slide, and then utilize them for downstream phenotyping and molecular analysis. And so at the bottom of this page, you see where our product line that I'll tell you about in a little bit cuts across, so the basic research through the translational phase into potential clinical diagnostics. So this is the overview of the product platform, the portfolio. This one is a little bit focused on the cell-based work, but I'll try to connect it to tissue also in a few moments. So we have a sample preparation system that's called AccuCyte. And this is basically a very simple centrifugation-based system that collects all nucleated cells in patient blood. It doesn't select on the basis of any biomarker or size or shape. So it really collects everything that's there. So its recovery and its sensitivity is incredibly high. And then once collected, we spread onto a standard microscopy slide and then do staining and imaging. The staining is done by the RareCyte staining kits. Now, these have up to 7 parameters available. That's also true for the tissue work that we're doing. So in terms of the instrumentation, 7 channels available. So in the case of liquid biopsy, we use 3 for the cancer cell, and then we have up to 4 open channels to phenotype those cells. And then there are 2 models of instrument, as you can see here, and they're split really by the usage of them, I would say. They're very similar in terms of capability, but we have one that's a high throughput device that would typically be found in a core lab type scenario that can handle up to 80 slides in an automated fashion. Everything is barcode driven, so it's LIMS compatible. So in terms of sample tracking and sample management, it's directly integratable into LIMS systems. And then the second system is the CyteFinder 2 with CytePicker, which is our needle-based picking device for microregion retrieval of tissue or for single-cell retrieval of liquid biopsy samples. And you know that becomes so critically important because we have a really strong belief that protein analysis and molecular analysis are complementary. It's not one or the other. You gain different information. And as I'll show you in a data set a little bit later, that information is critically important both in drug development but also patient diagnosis. So thinking about the liquid biopsy workflow first, how do those products fit into the workflow? So in traditional pathology, it's collect the sample, make the block, cut the sample, put the sample on a slide, the section on a slide, stain the slide, pathology review. This is very analogous to that, both in our liquid biopsy and then obviously in our tissue mode. So in this case, there's a blood draw from a patient. That would happen locally at a hospital or a remote site. There's then 3 days of stability where that sample can be either used or shipped somewhere, and particularly in a CRO type scenario where diagnostics are being performed, that shipment could be global, so this is quite an important feature. Processed onto a slide. Once on a slide, it's stable for at least a year, which means that those slides could be banked and stored while biomarkers were being discovered, while assays were being developed, while clinical trial processes were being configured. And so then those slides are available for those for that work. We then stain, we then scan and image, and then the output here is, in this case for cell-based work, it's a report, and that report uses machine learning a large number of inputs to score a cell as definitely a cancer cell, and maybe a cancer cell, or probably not a cancer cell. And that score is then used by a pathologist or a trained reviewer to quickly check through and say, okay, I confirm these scores. After that scoring process, there is then the opportunity for single-cell retrieval and analysis. And Kyla will actually talk a lot more about that later on and show you some data of work that we've performed. This is the only data set that I'm going to show you, but it's a really important one. It's work that was performed by the University of Washington. It was an independent study, and it's actually, I'm going to use it to make my point about the combination of protein and molecular being so valuable and impactful in terms of patient diagnosis. You're but obviously equally in research. So on the top graph, what you're seeing is enumeration of cancer cells in patient blood. This is a triple negative breast cancer patient, late stage. And so as you can see in the top chart, there's what we would say a very high number of cancer cells when that patient initially presented, over 1,000 cells per milliliter of blood. That patient then went through platinum-based treatment. There was a spike in the cancer cells in blood, presumably as the cells were being shed from the tumor. It then declined to a baseline. This was a monitoring over 50 weeks, and you can see there's a large number of samples. Obviously, this couldn't be done by traditional tissue biopsy. So the impact of liquid biopsy here is clear. That patient then began to progress. They then went through secondary treatment. You see the same sort of spike, and then a decline back to a baseline. What's shown in the bottom chart is aligned with the arrows in the top one, individual cells were picked from that patient's sample and then went through whole exome sequencing. And what you see is sort of susceptibility genes and oncogenes laid out side by side. And if you look very sort of globally at the data, the number of mutations are greatest when the number of CTCs in that patient blood are lowest, right? Suggesting that there is a greater number of variants occurring, when that patient is at their lowest CTC count. And therefore, the implication here is that these are probably and most likely the most dangerous of cancer cells in that patient that are constantly mutating and beginning the progression that we see in that patient over time. So combining the protein-based information that says, well, this patient looks in great shape for 30 weeks, with that molecular data says, well, hold on, there's something else going on here. So that was a data set on cell-based work. We're going to switch to tissue analysis. I described the workflow for tissue, for cells, and clearly it's very similar now for tissue. In this case, we're showing the use of the Ultiview Ultimapper kits. We like those kits. This could be any type of kit, but we're going to show you some great data with those kits. Staining on automated systems or via manual. Imaging on the CyteFinders, either one. Image visualization on our SiteHub software, which I'll talk about in a moment as a really powerful tool for interrogating data. Image analysis with Indica Labs, the Halo product, and then finally the option to retrieve individual microregions from tissue. So this slide really just shows you some of the features of the instrumentation. The two products that I mentioned, the CyteFinder 2 and the CyteFinder HT, separated by one at the moment has the picker, one doesn't. In our roadmap, we look at that as we suspect high volume picking is going to become a standard both in academia and in industry. And then some features in the middle that, I mentioned high resolution. These are, as you'll see, and I hope you'll agree, these are amazing quality images. It's a very fast scenario, typically around 10 minutes for a 1.5 by 1.5 millimeter section. So it's a very high throughput scenario. And then maybe the last thing to mention here is that and importantly for this integrated discussion this morning, these files are all available in terms of their file structure for use by other downstream softwares. There's no proprietary file structures in place. So I mentioned the SiteHub software. So this is a really, I think, for anyone working in a high volume lab, the impact of this shouldn't be underestimated. You generate large amounts of data, large image files, gigabytes that go to terabytes very quickly in terms of the overall number of data sets that you have. So the need for a really powerful, stable, and robust database to support those images is very strong. So in this case, we have SiteHub software that works with both cells for the liquid biopsy and tissue images. And this is a scenario where you can set up projects, have multiple users, everything is sort of password controlled. You set up your project, and then you can manage all of that information and data in that database. And in this first example, we just show a very nice image here that is in full screen, enables a user to move around that image and interrogate it deeply. And this really summarizes some of the features then that once you have that image, you can pan and zoom around it in digital pathology mode, looking for regions of interest, zooming in to find particular cells of interest. You can annotate those images with anything that you want. My name is Fred, whatever it might be. More importantly, some information about that cell type. And then you can use pseudo-colors in any particular format that you prefer. Once you have your sort of color panel defined, you can then utilize that across all of your images. You can share it with others. And one of the most important features here is that this is a server-based system where all of that information is accessed remotely or can be accessed remotely. So a scenario where a scientist is generating data, maybe working with a pathologist to review that data in different locations is readily achievable. In terms of the multiplexed imaging piece, this software supports comparison side by side of up to 6 images. So in terms of comparing colors and looking at different phenotypes, it's readily achievable using this system. So with that, I'm going to pass to Kyla, who's going to talk a little bit more about some imaging and show you some examples of the UltraView data sets.
Thank you. Thank you, Leighton. So I'm excited to take you through sort of some the tissue imaging workflow with some examples showing the UltiMapper IO kits from UltraView, and then some of the tissue picking work that we've done in collaboration with Peter Sorger's lab here, actually, through an STTR grant. So as Leighton alluded to, tissue has information at multiple levels, from the architectural level down to the cellular detail level, and finally at the molecular level from RNA and DNA expression. And the CyteFinder platform allows you to interrogate all 3 levels of information from the samples. So this is some CD3 T cells in lung cancer tissue. This is stained with the UltiMapper PD1 kit from UltraView. Some nice T cells. And we can start to, with multiplexed immunofluorescence, you can start to layer in other markers to learn more about these T cells. And so here you'll notice that some of the T cells are co-expressing the memory marker CD45RO. Start to learn a little bit more about the sample. Oops, what happened? And now we layer in the exhaustion marker PD1, and you'll start to notice that actually quite a few of these cells are showing T cell exhaustion. And then if we finally layer in cytokeratin and the nuclear stain, you start to see more of the structure of the lung cancer tissue at this point, and you'll notice that these T cells are infiltrating between the alveolar regions of the lung cancer. You start to ask more questions. What's happening here? Can I learn more about those individual T cells? Are the ones that are kind of invading into the cancer regions the same or different than the ones are in other parts of the tissue? So with that, I'll move on to what we call our Pick-Seq workflow. So the CyteFinder 2 imaging system has the integrated site picker retrieval module, which is actually just a little needle, sort of like a biopsy needle, that can get loaded into the instrument. So we have a little cassette, has a rack of needles in it, you load it in, the instrument comes and picks up the needle and automatically does the calibration. You take the rack out and put in your sample and you're ready to go. You will have at this point already done whole slide imaging of your sample and probably in the software reviewed those images and marked regions of interest that you wanted to go back for retrieval. Those coordinates get loaded back into the system, and you load the sample in, and using the needle you just say pick cell, and it will move the needle down and aspirate the cell off of the slide, and then transfer it to a PCR tube. The PCR tube, you can use any PCR tube, but RareCyte has developed a flat bottom version of the PCR tube, which allows us to image through the bottom so we can confirm the deposit of your cell into the PCR tube. This is just a nice confirmation. If you're going to take that into downstream molecular analysis, which can be quite expensive, it's nice to know that your sample made it in there. And so with transcriptomic analysis of samples, you can interrogate thousands of expression, thousands of levels of expression in those cells. So this is going to be just a proof of concept experiment we did using the tissue picking workflow. In this case, we started with FFPE tonsil tissue and stained it with a panel on one serial section. That was allowing us to find regions of interest for doing the picking. On the second section, serial section, we did just a brief nuclear stain and then retrieved regions of interest from the serial section. And then we did RNA-seq analysis on those retrieved sections, which then allowed us to discover something about the sample and confirm the RNA-seq findings on a third section. And the reason that we don't do the retrieval directly from the stained sample is that the antigen retrieval seems to be incompatible with RNA quality. And so you can't do RNA-seq on samples that have undergone antigen retrieval. So here is just a quick whole slide image of a tonsil tissue that's stained with a really basic panel. We've got CD3 marking T cell zones and CD20 marking B cell follicles in the tonsil tissue. And we've identified several regions where we'd like to do cell retrieving. So here you can see we've used the coordinates from the first slide to go back to the same region on the serial section that was only stained with a nuclear marker, and we can retrieve samples from each of those sections. This was using a 40 micron needle, so each punch is 40 microns around. You get kind of 5 to 10 cells per pick. It's not a single cell pick. In this case, we're getting just a few. So we ran RNA-seq on those individual picked regions, and we get, after doing differential expression analysis and hierarchical clustering, we get kind of nice differential expression of the expected T cell and B cell markers. You can see that the T cell zones, here we go. You can see that the T cell zones are differentially expressed, the markers, than the B cell zones, which is nice and what we expected. We also ran this data through CyberSort, which looks at, it's a trained data set that looks for cellular type phenotypes in the RNA-seq data and tries to deconvolve which cell types were in the picks. And so we get kind of what we expect in the T cell zones. We get mostly T cells, and in the follicles you get primarily B cells. But you can see that there were probably also some other cell types in those picks. But because the RNA-seq data set is very large, we wanted to also convince ourselves that we weren't just seeing what we wanted to see. We ran a principal component analysis, which basically takes all of the thousands of data sets that you get from the RNA-seq and collapses it into a nice 2D plot. And on this plot, dots that cluster together will have similar transcriptomic expression levels. And so you'll see here that the T cell zones are nicely clustered together and are differentially expressed than the follicles. The one thing we weren't expecting to see was that the follicles actually cluster separately from each other, which says in transcriptomic space, these are actually very different. So that led us to do further investigation. And here we actually came up with several genes where we see, first one, we just confirmed that the T cell and the B cell zones differentially express this T cell marker, which we expect, but then here are some additional B cell markers where the two different follicles have slightly different expression levels. And I'll just draw your attention to the CD21 marker where follicle A has much stronger expression than follicle B. That led to what we call a Pixie informed panel, where we took the next serial section and we stained it with this new panel. And you can see that actually the staining replicates the RNA-seq expression data. So this is pretty exciting. We think that this could be used as a biomarker discovery tool. Now this is not, CD21 is not a very exciting biomarker to discover in tonsil tissue, but you could imagine doing this on a much more exploratory level. I should also point out that this panel was actually, we came up with the markers and the Harvard team had already had some experience using those markers in their hands, and they very rapidly threw together a list of off-the-shelf antibodies that we could use to try and stain it. And we had transferred the assay and done this staining in less than 2 weeks. So it was actually very rapid panel development, in part because of a lot of the work that the LSP has done developing a nice cohort of kind of biomarkers that they have qualified in tissue. So just in summary, the CyteFinder 2 uniquely enables discovery at all 3 levels of information that you can achieve in tonsil tissue or in FFPE tissue and any type of tissue, really. And with that, I'd just like to give a quick acknowledgement to the LSP for their collaboration work. They helped us with a lot of the sequencing analysis for the Pixie experiment that I demonstrated. And this was done on a grant, an STTR grant that we are jointly doing with Peter's lab.